So this is a pleasure to actually present to you. Our lab started about 2 years ago. And what we are generally interested in is understanding how disordered systems are relevant to the propagation of disease. And we try to look at this sort of class of proteins by looking at through different lenses. And I'm going to try to explain to you what I mean with that. But firstly, I just want to give an introduction as to what are disordered systems. So they're very important because they actually form 30 to 50% of our genome. So this is either partially or fully disordered. And if we think about how they're different to normally what we think about folded systems like proteins, here I'm showing you sort of a textbook representation of a protein landscape where you can see that there's an energy minimum formed, and that's sort of thought to be the energy favorable state of the protein that can adopt accordingly its function. Now, with disordered systems, actually the energy landscape is much more heterogeneous, where we see multiple fluctuations happening, and this gives rise to the intrinsic sort of flexibility and plasticity of this class of proteins. And as Lionel said, we are interested in looking at these because they form different states, physical states throughout the cell, some due to their multivalent interactions and forming sort of these mesoscale oligomeric structures that are relevant to disease. However, in functionally, they're also very important. And actually, you can see that they are very sort of a challenging class of proteins to work with because they're so heterogeneous that they can adopt multiple conformations. So conventional tools like confocal microscopy does not allow us to really capture all these different dynamics and heterogeneity of these systems. And thus, this is why we adopt lots of single molecule approaches to really be able to detect them. And also from the therapeutic point of view, they're also challenging because sort of structure-based drug design is very challenging for these because, of course, adopting different conformations is hard to find hits for these type of this class of proteins. And they're also present throughout the cell, so they are forming, they're involved in cell signaling and cell cycle regulation. They're part of the lots of cytoskeleton components. They form the barrier between the nucleus and the cytosol through the nuclear pore complex. And as I said, most recently, they're related to form phase separation and forming these biomolecular condensates, both in the cytosol as well as in the nucleus. And most relevant to our lab, they're also present in a lot of diseases, mainly focused on neurodegenerative diseases. And what's quite interesting is that for each of these neurodegenerative diseases, I'm showing you here 4 of these, there is a disordered system that's been associated to it. And this disordered system will form different species that result in the formation of what are known as their hallmarks, which are aggregated proteins. And these are sort of patient samples where you can see like plaques of these proteins that are formed. And from our lab, what we are mainly interested in is understanding, of course, how these plaques are formed, but also understanding how and why they propagate in different pathways. Here in these different colors, you can see that each of these pathologies, the propagation is very different. And what really defines the vulnerability of a cell-to-cell contact that defines the pathway of these pathologies is what we're interested in. And what we think how this is happening is there's this imbalanced regulation of both proteins, but also lipids that are really giving rise to the onset of these diseases. And we're mainly focused on understanding the early onsets. And the way we sort of envision trying to target these different pathologies is by looking at it through the different lenses that I said in my title, where we want to be able to look at it at the nanoscale, so looking at molecule, sort of protein-protein interactions or protein-lipid interactions, but also moving for more looking at the oligomeric assemblies or sort of a crowding environment that these proteins undergo, especially with organelle contacts. I'm going to discuss with you. And then further on at the macro scale, sort of looking at cell-cell communication and tissue level organization. How do these actually propagate, and being able to define physical models that allow us to see how these systems actually find their propagation pathway. So I'm going to go through these little different scales and the tools that we use to understand these. Firstly, we heavily use fluorescence correlation spectroscopy, more on the sort of single molecule nanoscale level, where we're interested in using this tool to understand typically binding. So we can think of protein-protein interactions or protein-lipid interactions, although we also use it in the propagation models as I will describe later on. But this tool is, as you probably have heard in your workshop, is very good at being able to give you quite a robust and quantitative number of molecules, so concentrations, as well as it defines the diffusion time. So looking at the size of your particle, especially for these different disorder system undergoing oligomerization states, this is a nice tool to define these. We also use single molecule FRED, which is also based on diffusion, where we can look at conformational dynamics. So understand by putting a probe, two probes on the same protein or on different proteins, and understand this sort of the conformational dynamics our protein system might be undergoing. In terms of the sort of nanoscale to mesoscale imaging, we heavily use fluorescence lifetime imaging. We try to go away, as I mentioned, from confocal imaging, because this approach does not give us the quantitative nature that we would want. But here, just comparing a disordered system in our biological samples and with intensity and FLIM, you can see that while they, you have different intensities, actually they correspond to also different lifetimes. So they're clearly undergoing different states within a confined spot that they are observed in. So we're interested in understanding what these are. And of course, we know that FLIM can give us information regarding the crowding, but also sort of vesicular systems within the cell, like pH changes that we can monitor for these different systems. We try to go from the mesoscale to the more macro scale by looking at also more functional imaging. So we do perform a lot of calcium-based intensity imaging for understanding some more neuronal network activities. But by FLIM, we can also look at in the sort of single cell level, looking at specific calcium concentrate, localized calcium concentrations within the cell, and also look at how that correlates with our protein here shown in white. That is accumulating in these single cell level resolutions. With functional imaging, we can also go to the more full tissue imaging. We perform metabolic imaging on these here. I'm just showing you what metabolic imaging can do, of course, but in our case, we're not really working on cancer cells, but more on full organoids. These are brain organoids that I will go into in a bit, that we can look at how cell-to-cell differences in metabolic changes are occurring. And this sort of FLIM is one of the main approaches that we use because it allows us to be quantitative in sort of this complex environment that these neurodegenerative diseases are found and these proteins are found in. So in the lab, we've got sort of these different interest groups. What I really want to point out, though we think about these diseases both as proteinopathies, but we really think that they're also linked with changes in lipid landscapes. And that's what I will mainly focus on today. I'm going to go through hopefully like two case studies that we've been working on in the last 2 years and sort of the application. We also are interested in developing new tools and sort of imaging methods that allow us to really underpin the complexity of these diseases. And if I have not already told you how complex disordered systems is, I will do by showing you here what Alzheimer's disease looks like and how complex this single disease is. Where you can see that here this is a nice representation of all the genetic risk factors that have been associated with AD, and that each of these risk factors is associated to a cellular process. And what we think is that one maybe sort of aspect that has hindered the field so far is that a lot of these different processes have been looked at in a single way. So we either focus on protein aggregation or lipid metabolism. And what we're trying to do is to combine these and see where is there the coalescence of these different cellular pathways that are being affected in disease. And that's mainly what we're trying to address. So moving on to the first case study on Alzheimer's disease, what we are interested in is understanding not only how the protein is affected in a single cell, so in neurons, but also in astrocytes. And why we care about this is because actually one of the early onsets of Alzheimer's disease is failure in lipid metabolism. And the main aspect and the main sort of mechanism of lipid homeostasis in the brain is happening between neurons and astrocytes. So this is a bidirectional mechanism where astrocytes are there to provide metabolic support to neurons. And on the other hand, neurons need a lot of lipids in order to be able to take them to the synapses for synaptic recycling and transmission. But then neurons themselves are really bad at actually catabolizing lipids. So if there's a buildup of lipids, what the neurons do will be to secrete them out, and then the astrocytes are there to actually clear out the excess lipids in neurons. And the main sort of trafficking component in this is what is known as ApoE. And this is a apolipoparticle. So it's a protein that is on these vesicles. And these are produced in astrocytes and they're secreted, and the neurons will take it up and accomplish their cholesterol need, for example. And what's quite interesting with ApoEs is that there are 3 different alleles of ApoE, and the majority of us is ApoE3. However, in both ApoE3 and 2, which is really a minority, are thought to be protective, whereas ApoE4 is a major risk factor of Alzheimer's disease. And we're trying to understand how that might be the case. And sort of combining this lipid and protein aspects together, the other key component of Alzheimer's disease and a sort of key phenotype that we know of is the tau protein. Tau is generally expressed in neurons, and its main function is thought to be on microtubule binding. However, in the recent years, we've been coming to understand that actually tau does not only have a life in the neuron, but actually it's being secreted out. And that brings me to that propagation model. So what we think might be happening is that tau, while expressed in neurons, inside the neurons, is secreted at some point and then propagating to the next neuron or the next cell, which does not have to be a neuron, but potentially an astrocyte. And so what we're trying to see is the connection between the protein aspect in Alzheimer's disease and lipid aspect, and the combination of the two. And what my postdoc in the lab, Anna, is doing is looking at this at the 2D level, so at the single cell level, but then also building these single molecule tools on the more advanced organoid, more complex organoid model. So what organoids are is basically stem cells that we differentiate in such a way that we can get sort of small brains representatives for understanding these diseases. And of course, we cannot age the brain, but it is a good model to studying early onsets of disease. And what we are thinking is that what we are currently seeing, like our initial observation, is that tau and ApoE are accumulating in these organoids, and they're actually sequestering in lipid droplets that really sort of halts the availability of lipids in neurons. And in order to first see how our system is, what we first wanted to do is understand, can we use these organoids as a propagation model? And in order to do that, what we initially did is here I'm showing you a organoid, and in white, what you're seeing is a calbrite. It's just a calcium indicator. In this case, I'm just using it as to show you where the cell bodies and the somas of the neurons are here in white. And these are the projections of these neurons. And here in red is tau that we've labeled. We've initially expressed it from bacteria, and then we've labeled it with cysteine labeling, and then inject it into the brain organoid. And this is at 24 hours, we can see a local distribution. And then after 7 days, we see that actually the tau propagates throughout the organoid. So this is a good system for us to actually look at propagation of these systems, of these disordered systems. Now, as I mentioned earlier, we use FCS a lot to look at binding. So we've used it to look at the interaction between ApoE alone and in the presence of tau. So we can definitely see that they are binding before even internalizing in these organoids. But then we also use FCS in a sort of a bit non-conventional way to understand the kinetics of uptake. So what we do is we have tau and we have apoes in our focal volume, but then we put our focal volume in solution and measure over time the number of molecules. So we can look at this by looking at what's the drop in the number of molecules over time, so we can measure the kinetics of uptake, but we can also look at secretion, because as I mentioned, these proteins are expressed inside the neurons, but then they're secreted. And by using the number of molecules and the quantitative aspect of FCS, we can also look at secretion of these molecules. So this sort of allows us to be quantitative in our propagation models. And by the use of FCS, of course, we can also monitor oligomeric state. So what is actually being secreted is a monomer or an oligomer. That is what we are observing. So while we saw for the sort of the progression of this protein in these tissue-like systems, we also monitored how these are different in different cell types. And one thing that we really were able to see is that tau here in red is in neurons has a very much more dynamic state compared to when we compare it to astrocytes. So there's differences, and this is the complexity of this type of proteins, that they're different in different cell types. So there's a cell type specificity that makes these important. And also, if we look at it by the use of FLIM, we can see that in neurons compared to astrocytes, and this is I'm using a FRET-labeled tau protein in this case, where we can see that in neurons, actually, this same construct has a different conformation compared to when we are looking at astrocytes. So there is a very cell-specific nature of this. We combine this with EM-based studies as well, and you can really tell, like, with EM that actually this protein is forming this sort of fuzzy-like structure, more ordered structure here, more like tangles here, and in the cytosol more like sort of string-like structures that we're seeing, and we're trying to identify what these actual structures are. One thing that we've observed is that, in comparing again neurons to astrocytes, is that in neurons, the dynamic nature of the tau that we saw correlates with a lot of lipid component in there. And one thing that you can see here is that I'm staining with body P, and we can see that the tau particles that we see are correlating with the lipid neutral lipid dye that body P stains for. Whereas in astrocytes, this is not the case. We don't see the dynamics and we don't see the lipid components in there. So we really think that these are the tau and lipid dynamics is very important for the nature of tau in neurons. Just looking at the propagation, tau, as I mentioned earlier, will enter inside the somas of the neurons. We can see that very clearly. ApoE, on the other hand, doesn't enter so efficiently. However, when we add ApoE and tau together, tau seems to be cargoing ApoE inside, and we can see really nice co-localization of the two particles together. So we've quantified this as well. We were able to quantify this, and we can definitely see that there's generally more tau accumulating, more ApoE accumulating in the presence of tau, and this is also allele specific, comparing ApoE3 and ApoE4. And also the dynamics is changing, where we see that ApoE3, which is, again, just to remind you, we think that's more of a protective nature compared to ApoE4, is less dynamic compared to ApoE4 and plus tau in the neurons. And what we've noticed also over time is this here, I'm showing you organoids that have been incubated with both tau and ApoE at 48 hours, 5 days, and 15 days. We see that, well, the dynamics is changing, and we see the buildup of body P over time. So there's clearly some sort of sequestering of lipids happening. And we're able to image this nicely, where we can see that in these lipid droplets that are stained with body P, we see tau accumulating and ApoE forming this ring-like structure around it. So this is where sort of we think that there is accumulation of these in these lipid droplets. Okay, so I'm going to try quickly to go through the second case study, which is on alpha-synuclein. And alpha-synuclein is also a disordered system implicated in Parkinson's disease, and also aggregates. And one recently, it's been shown actually to aggregate in these, what they're known as Lewy bodies. And while generally we always thought that these Lewy bodies were made out of aggregated synuclein, it's coming to our understanding that it's actually not just protein, but a lot of lipid components that are also present in there, including mitochondrial fragments, and also lysosomes, ER fragments that are building up these what are known as Lewy bodies. And this is not surprising, because actually in Parkinson's disease, an alpha-synuclein has been associated to multiple times to mitochondrial dysfunction, ER, and lysosomal dysfunction. So this is not so surprising. However, we've never really seen synuclein localized really to a single organelle. And one theory that we have is that it's not a single organelle, but rather it's an interorganelle contact site that these proteins are localizing with. But of course, studying interorganelle contact sites are very challenging. And one way we're trying to do this is firstly trying to identify really if it is on the organelles, in the interorganelles. And what my PhD student, Elisabeth, has done is look at trying to express two proteins, one in the ER and one on the mitochondria, that by co-expressing these two, you enhance the effect of having these interorganelle contact sites between the mitochondria and the ER. And what you can see is after the addition of synuclein in these cells, we see that these are really that synuclein localizes in these contact sites. And what our theory here is that these interorganelle contact sites are the platforms that drive actual pathology and result in these Lewy body accumulations that we're seeing. And in order to look at this a little bit more in terms of the mitochondrial dynamics and understanding how synuclein can be implicated there, what we're doing is trying to do time-lapse imaging of the mitochondria, both in media as well as in hypotonic solution. What the hypotonic solution does is actually starts vesiculating your organelles. And from the imaging perspective, this is really nice, because then we can actually start seeing full organelles and sort of the lumen and the membrane outside. So from the sort of the resolution side, that's also very nice, but also it gives us a functional assay, because if we add synuclein, what we actually see, vesiculation happens, but what is really happening is there is a crumbling of these organelles. We don't see so much nice round vesicles happening, but rather this sort of sequestration of organelles. And we compared this with different also disease mutations of synuclein, as well as truncations of synuclein. And depending on the variant, you see morphologically that the mitochondria are undergoing hypotonic swelling, but to different extents. And so we think that synuclein has an effect on how the dynamics of mitochondria at these interorganelle contact sites behaves in the presence of our protein. And we're currently trying to understand this. To do so, we are also adopting expansion microscopy to be able to really expand these interorganelle contact sites to be able to visualize our protein. And at this stage, we are coming to about the 16x expansion that has been recently shown. So we were able to adopt that. Now we want to be able to do this in the presence of swelling as well. So we can actually really see how is our protein affecting the mitochondrial the mitochondria in the sense of swelling, if it's really disrupting its dynamics, and being able to see if we can adopt a sort of or address a function of synuclein in these interorganelle contact sites. We're also combining expansion microscopy with FLIM to be able to get the sort of the quantitative aspects that we want in this way. And lastly, this is the last slide, I promise. What we also been looking at is is adopting tools. For example, one big limitation with multiplexing FLIM is the fact that we don't have good probes in the near infrared, or at least with a good lifetime that we wish to have. And one thing that we've adopted a lot in our systems that we are imaging is the use of SNAP and HALO. And we recently published a paper where we've showing that actually in the presence, when we add ATO 700 to halo or snap, we actually increase the lifetime, and we reach a lifetime of about 2.2, which is relatively good, in the sense of it's in the range of GFP. So we are trying to work and see how we can get better probes in the near infrared, and this is in collaboration the Brioche Hagen lab in Berlin in FMP that we're working on. And with that, I'd like to thank my lab